Today's review session is going to be on cell energy, so we're going to be focusing on respiration and photosynthesis. So let's just start with a review of the words catabolic and anabolic. So catabolic are pathways that release energy by breaking down complex molecules into simpler compounds. So this would be an example of what happens during respiration when glucose gets broken down. That would be a catabolic pathway because energy is being released. An anabolic pathway is when energy is consumed in order to build complex molecules from simpler ones. So that is what we see during photosynthesis when we have to use the sunlight energy to form the glucose molecule. So basically how this works is ATP powers cellular work by coupling exergonic reactions to endergonic reactions. So a cell has to do three different main kinds of work. A cell can do mechanical work, transport work, or chemical work. And to do these jobs, the cell is going to manage the energy resources by energy coupling. So basically, the use of an exergonic or an energy-releasing reaction will be a process to drive the endergonic or energy-absorbing action. So exergonic, exer means exit, so energy-releasing, and that energy that is released is then going to be used to provide the energy to allow the endergonic, where energy is absorbed, to take place. So they work together hand in hand, which makes sense on how that photosynthesis and cellular respiration works hand in hand like that. Another thing to keep in mind is that enzymes are going to be used to help speed up reactions by lowering that energy barrier or by lowering the activation energy necessary to make a reaction take place. So remember that a catalyst is a type of protein. It's going to be an enzyme that is a catalytic protein, and it's going to speed up the reaction if it does not get used up by the reaction. Okay, so catalysts do not get used up in the reaction, so they can be used over and over again. So an example of an enzyme used or catalyst that's used during a reaction would be the hydrolysis of sucrose. So remember, hydro is water, lysis is to break apart. So by the breaking apart of sucrose, by using water, we insert water into it to break it apart into its smaller components. We use the enzyme sucrase, and that is going to be a catalyst that helps to make that breakdown of sucrose happen faster and with less energy. So let's talk a little bit about the enzymes. So enzymes are substrate specific, which means that they will fit into a specific substrate. So the substrate is basically the reactant of the chemical reaction that the enzyme is going to act on, and it's going to bind to that substrate or reactant into a specific binding pattern. So specific enzymes work with specific substrates based on their binding and being able to bind to the substrate. And so when they bind to the substrate, it's called an enzyme-substrate complex. And the site where the enzyme actually binds is considered the active site. So over here in the first picture, you have your enzyme, which is this little red complex, and we have, or I'm sorry, actually the enzyme is the enzyme is there, and then when you can see that it binds into that substrate in a specific location, and it fits perfectly, okay? So what happens is it lowers the activation energy that's necessary for the reaction. So in this graph we can see the black line here is showing you the amount of energy that's necessary way up here, okay, to get this reaction to take place in order for the reactants to become the products. Whereas with the enzyme added, it brings that amount of energy necessary down by quite a bit. Okay, almost decreases it in this particular example by half. And so it still results in the same ending, the same products, but is able to be done with less energy used, so therefore it happens much quicker. A cofactor is going to be a non-organic enzyme helper. So it's usually a mineral like zinc or iron that is going to be necessary for the enzyme to work properly, and they work together. So a cofactor is any non-organic helper that is going to be necessary for the enzyme to work correctly. A coenzyme, on the other hand, is organic. Okay, so it tends to be another type of organic compound, usually a vitamin of some sort, and they're also required for the reaction to take place. So a coenzyme works with the enzyme as well and is necessary for them to take place. So an example would be NAD or the coenzyme A. On the opposite side of that, there are also what are called enzyme inhibitors. And enzyme inhibitors keep the enzyme from being able to connect to the substrate so that the reaction cannot take place. So we look in A here, we have our enzyme is the big purple, and then we have the substrate is the blue, and it fits perfectly into that little active site so that it can react together and use, or I'm sorry, so that substrate can be use the enzyme to react as needed. So that would be the normal binding. When we have an inhibitor, the inhibitor will bind into that active site and block it so that the substrate can no longer get there. And so it stops that reaction from being able to take place, or at least stops it from being able to take place with the smaller energy we were looking for. There are also some inhibitors that aren't necessarily going to inhibit the actual reaction. They're non-competitive, and so they will actually fit down in another binding site, but it doesn't block the actual enzyme from connecting. So that is considered a non-competitive inhibition. So with that non-competitive inhibitor there, it will also keep the substrate and enzyme from reacting as necessary, but it's not considered competitive because it's not in the same active site. Whereas when it's a competitive inhibitor, it actually is blocking it from that active site specifically. In a feedback inhibition, this is another thing that it would be an inhibition. This is how feedback loops are sometimes stopped to keep from continuing on and on. Basically, the product that ends up at the end of the actual reaction becomes what shuts down the pathway. So we can see over here, we have our initial substrate up here, and it's called threonine. Threonine binds to the enzyme here on the active site, so that the reaction can take place, and it goes through a series of different reactions. You can see that it's got a couple different enzymes that are going to help it get through these different levels of reactions until finally it becomes the end product of isoleucine. When isoleucine is produced, isoleucine will now actually bind to that enzyme that was originally used for this reaction at the beginning, and by binding there, you can see it binds right here, it actually blocks and changes the shape of that enzyme, so it keeps the enzyme from being able to block to threonine again. So it stops the pathway and stops that feedback loop. When that isoleucine gets used up, as you can see up here, when it gets used up, so it comes off of there because it's being used, now it opens that inhibition spot up, and so the enzyme resumes its normal shape and can bind again so that more isoleucine can be made. So that's an example of a feedback inhibition that might take place, and how we can have the products becoming the inhibitor to stop a feedback until that product is necessary again because it's been used up. All right, so now let's move on to cellular respiration, which again was one of the catabolic energy or reactions, because we're taking a large complex and we're going to actually break it down for energy, so we're releasing energy. So energy is released as the electrons fall, and I use that word loosely, from the molecules to oxygen. So that's where the electron transport takes place. So the main overall steps is you have your food, which is the glucose, NADH, which is a energy transferring compound, the electron transport chain, that's what ETC stands for, not etc., but electron transport chain, and then the oxygen, which is the final acceptor of the electrons. And by getting to that point is where the energy gets released. So coenzyme NAD plus is the electron acceptor, and it's going to pick up some electrons and hydrogen as well, which remember hydrogen ions are basically just protons. And now it becomes NADH. So the H is representing that hydrogen proton that's been picked up. Those hydrogen protons have been picked up, and it's now storing those excess electrons because it was an ion, but now it's going to be storing it. Then the NADH carries those electrons to the transport chain, and then that transport chain is going to transfer those electrons to oxygen to make water, and in that process, energy is released from that transferring. Okay, so let's look at it a little bit with the diagram. Makes a little bit more sense here. So there's in cellular respiration that involves oxygen, and it's key to remember that oxygen is necessary to do the entire process of cellular respiration that we see in this picture. We will start with glycolysis, which happens outside of the mitochondria. It happens in the cytoplasm, and this is where the glucose is converted into the pyruvate. And in this process, we get 2 ATP produced, and electrons are going to start to shuttle from that process of breaking down glucose into the pyruvate. We're going to get some electrons added to the NAD plus to the NADH. So remember, this NAD these are the electron carriers. So we've got some electrons being carried here into the mitochondria from this initial process, and then the pyruvate are also going to enter into the mitochondria at the end of this process. And this only happens if oxygen's available. If oxygen's not available, then this will not go into the mitochondria here, okay? From there, we're going to get ready to start the Krebs cycle. At the Krebs cycle, in the process of moving to the Krebs cycle, we've got some more electrons being taken into the NADH as 2 acetyl coenzyme A's are formed. And in the Krebs cycle, we are going to continue to break down that pyruvate, and in doing so, we get some more NADH, so some more electrons being pulled off. And also, we can get a couple of ATP from this process as well, okay? And then finally, all of these NADHs and also this FADH, which is another energy carrying compound, are going to continue into the electron transport chain, where we're going to have some oxidative phosphorylation, in which basically the transferring of those electrons to the oxygen, and this is where the oxygen is necessary, results in us getting about 34 more ATP produced from all of those electrons that have been transferred over and through that electron transport chain. So per 1 glucose, we can get about 38 ATP. Now keep in mind that during this process, you can see we, right before the Krebs cycle, if we could follow that all the way down here, there is usually about 2 ATP used up in that process to make this happen. So the net tends to be only about 36 ATP, but overall about 38 are produced. But because 2 tend to get used up in the process right at this point, we say that the net would only be 36. Okay? All right, but again, oxygen's the key here, because without the oxygen, then this last electron transport chain cannot take place, and this is where the majority of those electrons get transferred, and where the majority of the ATP is able to be formed from that transfer. Is the energy from those electrons is what's used to be able to bond the phosphate to ADP to form the ATP. So inside the mitochondria, just so you kind of know where these 2 different processes take place after the glycolysis in the cytoplasm, the citric acid cycle happens inside the inner membrane here in the matrix, and the electron transport chain happens in this inner membrane, on the inner membrane fold. So it kind of happens across that membrane there. Okay? All right, so with glycolysis, again, that's the first part where the glucose first initially gets broken down, and we mentioned that oxygen has to be available in order for it to continue into the mitochondria, okay? So that would be this route over here where we have the full respiration take place where we release the electrons using the oxygen. It happens in the mitochondria. Again, oxygen is required because it's the final electron acceptor. And in the process of this, we produce carbon dioxide as a byproduct. Water gets produced where the oxygen gets added to those hydrogen protons from those energy compounds, NADH and FADH, and possibly up to 38 ATP, a net of 36. On the other hand, we know that oxygen's not always going to be available to our cells, not because we're not breathing, but because as we're breathing, the cell has a storage of oxygen. So the oxygen, when you're breathing in, goes into your blood, into the red blood cells. And so as our oxygen depletes within a cell, that forces the diffusion of the oxygen out of the blood into the actual cell. But when we're initially working out or doing things, we have a storage that we rely on to get going, and eventually that will run out, and we have to wait to, for the blood to bring the oxygen to the cells to kind of replenish it. So sometimes we still need energy, though, for those cells to continue to do work. So what happens is fermentation, and this is the anaerobic part of respiration when oxygen's not available, so no oxygen here. Okay, so no oxygen. So this happens in both plants and animals. By the way, respiration altogether, even with oxygen, will happen in both plants and animals. That's a common misconception. A lot of times students want to think that photosynthesis only happens in plants, so cellular respiration can only happen in animals, and that's not the case. Cellular respiration has to happen in all organisms. With oxygen can happen in both plants and animals. Without oxygen can happen in both plants and animals, as well as bacteria and other things, because they don't have mitochondria, so they wouldn't use the oxygen level. Photosynthesis does only occur in plants or autotrophs, but the respiration, the breakdown of the glucose into ATP is going to happen in all types of organisms. So make sure you do not get stumped or duped by that misconception of that it's only in animals, because it's not. Okay, so I digress. Anyway, so when after glycolysis is done and we have that breakdown of glucose into the pyruvate, if there's no oxygen available anymore, then it's going to stay in the cytosol. It's going to stay in that cytoplasm, and glycolysis basically kind of continues and creates either alcohol or lactic acid, because we need to be able to continue to break down that pyruvate, that process. So there are two different kinds of fermentation. We have alcohol fermentation or lactic acid fermentation. In the alcoholic, we have the pyruvate getting broken down into ethanol, which is a type of alcohol, so that's where the term alcoholic fermentation comes from. And then carbon dioxide is also a byproduct here. This is very common in some bacteria and also in yeast. And so we see this as a process that is actually used to our advantage when we do baking of bread. That's what helps make the bread rise, is the carbon dioxide from this process. It's also used in brewing of beer and wine because of the ethanol. Lactic acid fermentation is what we see mostly in human cells. Some fungi and bacteria will do this as well, but it's primarily an animal thing. Pyruvate is broken down into lactate acid, and this can also be used when talking about fungi and bacteria. We can use lactic acid fermentation in the process of making cheese and yogurt. Okay, it's also one of the things that very common in our muscle cells, and it builds up and tends to cause some soreness in the cells as well. So something to keep in mind as well is that we talk about respiration occurring with glucose, and that is true. It starts with that glucose, but many different types of organic compounds can be the starting point or the source of the fuel that can be broken down into and be used. So we can use carbohydrates, we can use fats, we can use proteins. All of those things can be used as fuel for cellular respiration. The one that we prefer to use is the carbohydrate, because the carbohydrate, as you can see in this picture here, it's kind of small, I apologize for that. The carbohydrate is, of course, going to be the easiest one because it goes right down into the glucose. It gets broken down into its building block, which is glucose, and so on and so forth, and that's what the system's set up for. However, fats can be used. Now keep in mind, fats are not going to get turned into the glucose, okay? Follow the yellow lines and you can see that the glycerol from a fat, or the fatty acid from the fat, is going to come into this process of respiration at different points. So glycerol will come in after the glucose would be getting broken down. So it would actually come in and be broken into pyruvate in glycolysis, but it's not going to become glucose. It's just going to be the glycerol first that gets broken down a little bit and then turned into pyruvate. Fatty acids actually skip that part and will come into the process right before the Krebs cycle. Okay, proteins are also something that can be used. This is the last choice we like to use because proteins are ultimately what make up our body. They make up all of the functional units of our body. So if we're breaking down proteins to use for fuel, then we're ultimately breaking down ourselves. So that's not a good thing. But if necessary, we can do it. Remember that the amino acid is the building block of the protein. So those then can enter in at either the glycolysis level or at the citric acid level. But again, they're not going to get turned into glucose. That's a misconception as well. A lot of people think, oh, your fats can be, when we need to use energy from our fats, they'll get transferred into a carbohydrate. No, fats are different than carbohydrates. They're two completely different compounds. One cannot become the other one. So they can still be used, they're just going to be broken down as a fat. And once your carbohydrates are all used up, then the fats get used. So that's really, you can't, your body's going to use the carbohydrates first and foremost. So anybody trying to lose weight has to cut back on their carbs in order to force the body to use the fat instead and break down for energy. Okay? But this is just to let you know that they can all three be used. They just come into this process at different points in time, but they all are possibly used for fuel. All right, so now on to photosynthesis. With photosynthesis, just want to kind of cover real quickly where photosynthesis takes place. So it takes place inside the cell in the chloroplast. And typically the chloroplast the cells that are functioning in photosynthesis is the mesophyll. So you can see in this picture up here with the leaves, the mesophyll is just one of the layers typically that is found in your leaves of the tree or the leaves of the plant. Okay, so the chloroplast that are inside those cells in that particular area of the plant are going to be where photosynthesis takes place. And remember that the chlorophyll, which is that green pigment that is inside the chloroplast, is going to be the main place where we're going to have photosynthesis take place because that's the pigment that absorbs the majority of the sunlight. And then also involved in photosynthesis are the stomata. And the stomata are little pores that are on the leaf. So you can see those little pores here. They're on the surface of the leaf, and that's where the carbon dioxide can enter into the cell and where the oxygen can exit. All right, so with photosynthesis, if you break down that term, photo and then synthesis, synthesis means to put together, okay, and that's what's going to happen in the Calvin cycle. That's where the actual putting together of the food or the glucose is going to take place. And then the photo part is going to be the light that's required, which takes place in the light reactions. So there are two main parts to this process, the light reactions and then the Calvin cycle, okay, or the dark reactions. So inside the chloroplast, we have these stacks of little green, they look like coins, and those stacks are called grana, and the individual coins are called the thylakoid, and then within those little coins is a space that is called the thylakoid space, or also the lumen, okay? And so what happens in the light reactions, that's where the light is necessary, and also water. So the light reactions require water and require light. And there are two systems, the photosystem 2 and photosystem 1. and they're both going to be absorbing light through these thylakoid membranes and through the chlorophyll there. And in doing so, it's basically electron transport chains. They both are involved in electron transport chains in which they're moving electrons in order to gain energy from that light. So in this process, the water gets split during the light reaction. So water gets split so that that oxygen can get used as the final electron acceptor. So just like you saw in cellular respiration, the oxygen is the final electron acceptor, and so that we can get that energy from those electrons. And the hydrogen that was from the water is going to act as protons to be added onto the NADs. And so you're going to see it becoming NADPH, and ATP will then be able to form into ADP will be able to form into ATP. Okay, so the hydrogen from the water is acting as a proton that gets added. So protons are added to the NADPH, which is necessary for the Calvin cycle. And the electrons are transferred through the electron transport system in order to get enough energy for ATP to form so that there is energy to make the Calvin cycle go. Because remember, when we're putting together a molecule, energy is absorbed. So we need to have energy to make that happen. And so this is where that energy comes from. Okay, it's from that light, the electrons from the light energy. Okay, all right. So oxygen gets released in this process of the light reactions, and the hydrogen protons that are now left over from the water are going to get transferred over into the Calvin cycle as a part of the NADPH. So the Calvin cycle is where the carbon dioxide comes into play. C for Calvin, C for Calvin cycle, C for carbon dioxide. So the carbon dioxide comes into place, and this is where the organic carbon and oxygen come from in order to form our food product. And so there's actually going to be a necessary of 3 carbon dioxide molecules to enter into the Calvin cycle. They enter one at a time, and at the end of each transfer through this cycle, we are going to end up with a molecule of not necessarily glucose, but a carbon like glucose. That's not the word I'm wanting. With a sugar molecule of some sort that will eventually become the glucose. Okay, so you get 3 of those. So let me just kind of show. I think I've got that on the. Nope, I didn't. Yeah. So, okay. So you're going to get, so it's going to be 3 carbon dioxides, one at a time, and at the end of each one we get the organic molecule. So you get 3 of those, and that's what eventually becomes that glucose molecule. And then here you're seeing it as sucrose, and that's just what would happen if you were going to store it. In plants, remember that glucose gets stored, and so that's one of the as starch and things. So, okay. All right. So that is the photosynthesis. So if we want to compare photosynthesis with respiration, again, respiration takes place in both plants and animals. Photosynthesis would be just plants. Respiration needs oxygen and food or glucose. Photosynthesis needs carbon dioxide and water and sunlight. In respiration, we produce the carbon dioxide and water and energy. In photosynthesis, we produce glucose, oxygen, and energy. Excuse me. The respiration takes place in the mitochondrial membrane in the matrix. Primarily, that's where the oxygen portion takes place. So that's why that's what's mentioned here, because this type of respiration is assuming oxygen is available. Photosynthesis is going to take place in the chloroplast. And then I've shown you here the 2 different reaction chemical equations. So you got glucose plus oxygen yielding water, carbon dioxide, and energy in respiration, and that energy is ATP. And then in photosynthesis, we have water plus carbon dioxide plus light energy. So that's light. Okay, so that's light energy, and that forms our glucose, and oxygen is given off. So photosynthesis, again, is an anabolic chemical reaction because energy is required to combine to make the complex compound. And then respiration, on the other hand, is catabolic because it is breaking down the compound, so energy is released. Okay. All right. So now on to some practice problems. So if we take a look at this problem, these first three problems are going to refer to this background information and the data that's given here. So we've got an experiment that's measuring the rate of respiration in crickets and mice at 10 degrees Celsius and 25 degrees Celsius. It was performed using a respirometer, which is an apparatus that measures changes in gas volume. Respiration was measured in milliliters of O2 consumed per gram of organism over several 5-minute trials, and the following data were obtained. So we have the mouse at 10 degrees, a mouse at 25, a cricket at 10, and a cricket at 25, and their average respiration rate is listed. Okay? So during aerobic cellular respiration, oxygen gas is consumed at the same rate as carbon dioxide gas is produced. So we have oxygen gas being consumed and carbon dioxide is produced. In order to provide accurate volumetric measurements of the oxygen gas consumption, the experimental setup should include which of the following? So remember in this information here, it told you that they're collecting it using a respirometer, which measures a change in gas volume. So it's not specific to the type of gas. It's just measuring any change in gas volume. So if carbon dioxide gas is being produced and oxygen gas is being consumed is going to be a change, and being produced is going to be a change. So in order to only get oxygen gas being consumed, we want to make sure that we have something that removes the carbon dioxide so it's not being used or it's not being mistaken in that respirometer apparatus. So must have a substance that removes carbon dioxide gas. Otherwise, a change in the amount of carbon dioxide gas will be picked up by the respirometer, and ultimately they would cancel each other out. So there has to be something in the setup that removes that carbon dioxide gas so that the only thing being measured by the respirometer is that oxygen gas being consumed. Okay? All right, the next question, still dealing with that same data. It says, according to the data, the mice at 10 degrees Celsius demonstrated greater oxygen consumption, so it used up more oxygen per gram of tissue than did the mice at 25 degrees Celsius. This is most likely explained by which of the following statements? So is it A, the mice at 10 degrees Celsius had a higher rate of ATP production than the mice at 25 degrees Celsius? So if they had a higher rate of ATP production, that means that they are using a lot of energy, okay, or needing a lot of energy. So the 10 degrees would need more energy than the 25. All right, let's think about that. B, the mice at 10 degrees Celsius had a lower metabolic rate than the mice at 25 degrees Celsius. Well, the lower temperature is going to be harder to maintain your body temperature. You got to remember, a mouse is like humans as far as being warm-blooded. So it's going to, the temperature is going to affect their temperature control interiorly. So if they are at a colder temperature, their body's probably going to be working harder to try to maintain temperature than somebody at 25. So I don't think that it would be lower metabolic rate, it would probably be higher. At C, the mice at 25 degrees Celsius weighed less than the mice at 10 degrees Celsius. That's not really going to play a role in their oxygen consumption. So I don't think that that would be pertinent answer. And then D, the mice at 25 degrees Celsius were more active than the mice at 10 degrees Celsius. Again, although that could make a difference in oxygen consumption, we don't have enough data, and the first part of this says according to the data, okay? And then it does say what's most likely explained, but I don't think that that's enough information to be able to say for sure that that's what it is, because most likely in the experiment they're keeping everything pretty controlled. So the correct answer here is going to be A. The mice at 10 degrees Celsius had a higher rate of ATP production than the mice at 25%. Because in producing the ATP, remember we have to use oxygen in cellular respiration to do that. And so that's why the consumption of oxygen would be greater at the 10 degrees. And the reason why you would probably see this happening is, again, because of that colder temperature. Because of that colder temperature, the mice is probably having to maintain their temperature, which usually involves shaking, or not shaking, quivering, shivering, as well as movement, things like that, muscle contractions. All those things tend to increase your temperature, and so therefore that would increase the use of energy and ATP. All right. Next question here. According to the data, the crickets at 25 degrees Celsius have greater oxygen consumption per gram of tissue than do the crickets at 10. So this trend in oxygen consumption is the opposite of that we saw in the mice. The difference in trends in oxygen consumption among crickets and mice is due to their what? Relative size, mode of nutrition, mode of internal temperature regulation, or mode of ATP production. Well, it's definitely not D, because they're animals. ATP production is going to be the same. The other three are all possibilities, although nutrition would probably not play a role here, because there's nothing being affecting that. It all goes to glucose, basically the same process as far as the ATP production is concerned. So the only other would be the relative size and mode of internal temperature. And because temperature is one of the main variable here, that is going to be our answer. Mode of internal temperature regulation is going to be the difference, because as a cricket, it's going to have a different internal temperature regulation than the mice. So that is the reason for that. Okay. All right. Next question. Experimental evidence shows that the process of glycolysis is present and virtually identical in organisms from all three domains, archaea, bacteria, and eukarya. Which of the following hypotheses could be supported by this evidence? All organisms carry out glycolysis in the mitochondria? Absolutely not. We can cross that one off automatically. First of all, because it said all, and we know bacteria do not even have mitochondria. Secondly, glycolysis does not take place in the mitochondria to begin with. B, glycolysis is a universal energy-releasing process and therefore suggests a common ancestor for all forms of life. Possibly. That one could possibly be correct. C, across the three domains, all organisms depend solely on the process of anaerobic respiration for ATP production. Absolutely not, because we know that just ourselves, we use aerobic respiration, so all organisms depending solely on anaerobic is not possible. D, the presence of glycolysis as an energy-releasing process in all organisms suggests that convergent evolution occurred. So we haven't talked really about evolution yet, but convergent means that you have a separate ancestor but are in similar environments, so you tend to develop similar adaptations. I would definitely say that's not going to be the case if we're talking about all organisms in these three domains, because we all don't have similar characteristics. So definitely not there. So the correct answer that is left is B, which definitely makes sense, that there is a possibly common ancestor for all life forms, and glycolysis is a universal energy releasing process. Okay. All right, next question. The chemical reaction for photosynthesis is 6 carbon dioxide plus 12 waters plus light energy yields us glucose plus 6 oxygen plus 6 water. It's written a little bit differently than we tend to usually see. A lot of times we tend to only see 6 waters over here and then no waters on the other side, but it ultimately means the same thing. So this is saying if the input water is labeled with a radioactive isotope of oxygen, which would be oxygen 18, then the oxygen gas released as the reaction proceeds is also labeled with the radioactive isotope. So which of the following is the most likely explanation? So this is kind of testing what you remember about photosynthesis. So A says during the light reactions of photosynthesis, water is split, which is true. Light reactions is when water is split. The hydrogen atoms combine with carbon dioxide and oxygen gas is released. Not correct with that second part. Carbon dioxide and hydrogen atoms do not combine during the light reactions. B, during the light reactions of photosynthesis, water is split, yes. Removing electrons and protons, yes. And oxygen gas is released, yes. All of that is correct, so I'm going to go ahead and circle it, but let's go to the next one just to make sure. During the Calvin cycle, which is the dark reactions, water is split. Oh, I don't even have to read the rest of that because water is not split during the Calvin reactions. It's split during the light reactions. D, during the Calvin cycle, water is split. Again, I don't even need to read the rest of it because I know water is not split during the Calvin cycle. So, you know, hopefully me going through these and kind of showing you and kind of going through and showing you my thought process and going through these questions is helping you with, you know, breaking down these multiple choice questions. All right, and so one other question here, this is a free response. This one shows us that matter continuously cycles through an ecosystem, and a simplified carbon cycle is depicted here. So we've got carbon dioxide in the atmosphere, organic molecules over here, and it just kind of cycles through. So A wants you to identify the key metabolic process for step 1, where carbon dioxide becomes the organic molecules, and the key metabolic process for step 2, where the organic becomes the carbon dioxide. And briefly explain how each process promotes the movement of carbon through the cycle. For each process, your explanation should focus on the role of energy in the movement. So we have, it's a two-part question with two different areas that we're trying to refer to. So we can start with, and again, you would want to answer these in complete sentences. I'm going to just keep it short and sweet just because of space and time, but you would want to answer in complete sentences. But for A, we need to first identify, let's start with step 1. So step 1, we're going from carbon dioxide being in the atmosphere to an organic molecule, and it's a metabolic process. So we can't be talking about like burning of fossil fuels or sediment forming, things like that. Metabolic is something that happens in a living thing. So what metabolic process is going to take carbon dioxide and turn it into an organic molecule? We just talked about it today, and that would be photosynthesis. For step 1. Okay, so photosynthesis is how carbon gets from the atmosphere to the organic molecules. And it's done by the fixing of the carbon into the glucose molecule. And how is the energy, the role of the energy in the movement. So when we are fixing carbon into glucose molecules, we are going to be using energy to do that because it is because we are forming a compound. So the fixing of the carbon into glucose molecule using energy from sunlight, right? This is photosynthesis. We use the energy from the sunlight, so using energy from the sunlight. So this will be just a kind of an overview of what you possibly might say. And then for B, or well, not B, this is still A. For step 2, how does the organic molecule go back to carbon dioxide? Again, that's something we discussed today, and that would be the cellular respiration is how we get the organic molecule, and that is done by the breaking down of glucose through, well, we don't even have to say through, we already said it was cellular respiration. So breaking down of the glucose to release ATP energy, which also releases the carbon dioxide. Okay, so that's just again, giving you a little bit of overview. And then for B, we need to identify an organism that carries out both processes. So what would be an organism that does both photosynthesis and cellular respiration? Ant. Okay, or you could even maybe be more specific and put an oak tree. Okay, again, you want to use complete sentences here. So let's just kind of see, based on the rubric, how well this was. So for A, it says we get 1 point maximum for identifying the system, that's the correct process. So we know that 1 was photosynthesis, or you could have just put in the Calvin cycle part of photosynthesis. So let me just go back. Did we put photosynthesis for 1? Yes, we did. For 2, cellular respiration. Also would have taken the citric acid cycle or Krebs cycle, because those are the areas where the actual carbon dioxide gets produced. So we would have earned our 1 point for getting both of those correct, because notice it says and here. So you have to get both of those correct to get that 1 point. Then in the explanation, so for the photosynthesis, we would need to involve some terminology about carbon dioxide being fixed. So we said that carbon was fixed, and that the energy is using light energy, or you could have said ATP energy from the light reactions, because remember ATP is formed during the light reactions. So either one of those, but we said light energy, and forming the organic molecule. And we said that we fixed the carbon into the glucose, so that's the forming of the organic molecule using the energy from the sunlight. So we would have gotten that point. In cellular respiration, we would need to say that the organic molecule is hydrolyzed or broken down. So we say breaking down of glucose, so we got that part. And then uses energy for cellular processes such as growth or ATP production. So we could have put ATP production as what the energy is used for, where the energy goes. And we said to release ATP energy, so that's good. And the carbon output is the carbon dioxide. So we put at the end, releases the carbon dioxide. So we would have earned the points here too. So you could earn 2 points there. Oops, went back too far. 1 point for getting all of this correct for the photosynthesis, and 1 point correct for the respiration. So this would have been 2 points plus the 1 point there, so you would have gotten your 3 points for that particular part. And then for B, any of these would have been possible acceptable answers. So we put plants, so that was correct. We could have also put algae. You could have put a photosynthetic protist like a Euglena. You could have put a cyanobacterium or a CO2 fixing bacterium. You could have put a lichen. Could not have put a fungus, but you could have put lichen. So those are just some examples. Okay? So hopefully this is helping you a little bit. If nothing else, just going through some of those practice problems and the process of elimination is helping, and just kind of seeing how the 3 responses are graded and what types of things they're looking for. And we will be back here again next week and do 2 more sessions. And if you need any help or have any questions with anything, please make sure you contact me. Have a wonderful day.